Instead of doing boxes for our introduction to genetics, what I'm going to have you do is you can either draw this yourself with these words or you can print it. On Schoology, it's called Genetics Keywords. These are the different concepts or words that you should be able to recognize in word problems. So as you go through the video today, the next video, when you come across these things, go ahead and take a minute and then explain it. Or after the video, go back and fill them in. So when we look at genetics, Gregor Mendel is often called the father of genetics. And this is because he had a background. He actually went to the university where he studied math and physics. And then he worked in a monastery where he studied, he was in charge of the garden there. And so he happened to work with pea plants. And I'll talk more about this in class. But what he did was he basically figured out that there's traits that are dominant and recessive. He figured out basically the laws of genetics. And so when we talk about genetics, we talk about Mendelian genetics, which is pretty much what you learned in 9th grade. And then we talk about non-Mendelian genetics, exceptions to what Mendel figured out. So anyway, what Mendel did is he worked with pea plants, and pea plants have different characteristics. The one we're going to look at is flower color. So Mendel worked with true breeding purple flowers. And when you see the words true breeding, you're going to kind of think of homozygous. So if it breeds true, that means it's homozygous for a certain trait. And what he did is he took pure bred or true breeding purple flowers, and these are the parent generation or the pea generation, and he crossed them. When you see the X, that means they mated. And so in his first generation, which is called the F1, these are the offspring down here. All 4 of these boxes represent the offspring. And in reality, with the true breeding, all of the offspring would always be purple. So then he took purple flowers and he took white flowers. And again, the white flowers were also true breeding, which means homozygous for white flower color. So here's the P generation, and here are the F1. Now, when you look at the F1 for purple flowers, you can see they're all true breeding or homozygous, dominant. For the white flowers, again, all of the F1 are true breeding for the recessive or white flower color. So when we look at what he did is he took the parental generation and he crossed a true breeding purple and a true breeding white flower, and he found that the whole entire F1, was purple. So Mendel was like, what the heck, how did you have the white phenotype completely disappear? Well, most people would stop at the F1, at the first generation. But what Mendel did is he allowed them to self-pollinate. When you look at the inside of a flower, it has both male and female parts. And so the yellow pollen is actually plant sperm. And what it does is it goes onto the top. This part here is the female part. And so it lands on top of that and it can fertilize its own eggs. So he let the F1 self-fertilize. And what he found out was that 3 of the offspring, 75% of the offspring were purple, and 25% of the offspring had white flower color. Of the F2, this is now our second generation. So we have our P generation, our F1, and our F2. And when we look at our F2, we see a 3-to-1 ratio. What that means, when you do like a Punnett square, let me go ahead and just show you real fast. When you do a Punnett square and you get purple, purple, and purple to 1 white flower, what's that saying is for every 3 purple, you get 1 white. That's a 3-to-1 ratio. Okay, so when we look at this F1 offspring, though, are they true breeding? If they were true breeding, then all of the F2 would be purple. But what we see is we have this one flower that has white flowers. So no, these are not true breeding. These are actually called hybrids. The other word for hybrid is heterozygous. Heterozygous. Okay, so here's just a little picture showing how Mendel was very specific when he crossed his plants. He got the pollen from the male part of the plant and put it on the female part of the plant, keeping track of what the parent plants were so he could measure and record the offspring. So Mendel, why he's so awesome, we will talk about him some more in class because it's one of my favorite conversations. Okay, so now some other genetics vocabulary. You have phenotype. Phenotype is your like physical appearance, but phenotype can also be like what's inside of you. So for example, like your blood type. I am B positive, my favorite. So I'm B positive as my blood type. That would be considered my phenotype. Even though you can't see that I'm B positive, it's still considered a phenotype. Okay, then you have genotype, and genotype is what codes for your phenotype. Awesome. So now here are homologous chromosomes. This is not a sister chromatid. I'm sorry, these are not sister chromatids. These are two separate chromosomes that are not duplicated. Okay, so I'm going to separate them so that we can focus on them a little bit easier. And what we see here is I get one chromosome from my mom, one chromosome from my dad. And let's look at the gene for eye color. So here's one gene, eye color. So I'm looking at one trait. And how many copies of each gene do we have? So I have the one from my mom, and I have the one from my dad. So I have 2 copies of each gene. Oops. So when I look at these copies, though, one says brown eyes, one says blue eyes. They're both for eye color. Now, these different options, I have a brown eye, I have blue eye on my DNA. These different options are called alleles. Alleles are just all the different versions for a gene. So in those flowers with Mendel, the alleles were a purple allele or a white allele. When you look at eye color in humans, there's brown eye alleles, blue eye alleles, green eye alleles, the different options that we have. But regardless, let's take human eye color, brown eyes, green eyes, blue eyes. There's 3 different possibilities, 3 different alleles, but you only get 2, one from your mom and one from your dad on your homologous chromosomes. Okay, now when we write these alleles, this is what we write with our letters. These are what make up our genotype. So these 2 chromosomes would be considered a heterozygous set of chromosomes. So here, if you have 2 of the same alleles, you're considered homozygous. If you have 2 different alleles, you're considered heterozygous. And if you again have 2 of the same, homozygous. Now, if I were to tell you, though, hey, I'm homozygous for eye color, right now, you wouldn't know if I had brown eyes or blue eyes. Therefore, we have to clarify. This one here is homozygous dominant. And this blue one here is homozygous recessive. So if someone asks about homozygous, you would need to clarify homozygous recessive, homozygous dominant. Heterozygous, you just leave it like that. You don't say heterozygous dominant. Okay. Here we have, when we talk about genotype or phenotype, if I ask, hey, what's the genotype? You would answer me homozygous dominant, heterozygous. If I ask for phenotype, you'd say blonde, dimples, no freckles. Okay. And we write our alleles with letters. The dominant is going to be capital, and the lowercase represent our recessive alleles. Okay. So, when we look at Punnett squares, a Punnett square is cool because it's showing us the probability of what could happen between 2 individuals. So when we look at like a male, for example, a father who's maybe heterozygous for eye color. Well, when meiosis happens, here, let me just draw this. So here's two sister chromatids held together. So here's a big B and a big B, little b, little b. In class, these would represent your dark green and light green chromosomes on the pipe cleaners. Well, after meiosis happens, inside of the sperm, half of the sperm have the dominant allele, half of the sperm have the recessive allele. So here, we can look at, there's a 50-50 chance that a sperm is going to have a dominant allele, a 50-50 chance a sperm is going to have a recessive allele. And the same is true for the eggs. So right here, what a Punnett square is really showing us are all the different possibilities. So if this sperm gets into this egg, here's the offspring. If this sperm with the green, gets into this egg, it's a heterozygous offspring. If this B dominant allele gets with this egg, here's the offspring. And if this sperm here with the recessive fertilizes this egg with the recessive, here is the offspring. So when I look at this Punnett square, what I see is here there's a 75% chance that the offspring is going to have like brown eyes, for example. So big B stands for brown eyes, little b stands for blue eyes. So here there's a 75% chance that each time this couple gets pregnant, the baby will have brown eyes. And there's a 25% chance each time the couple gets pregnant that the baby will have blue eyes. I can also see that for every 3 brown eyes, there's 1 blue-eyed offspring. Okay. Oh, there we go. So here's an example problem. In roses, if having thorns is dominant to no thorns, the first thing you always want to do is write down what is dominant, what is recessive. And let's say you have a heterozygous mom crossed with the homozygous recessive dad. You set up your Punnett square. Now it doesn't matter if the mom goes here or like on the side or on the top. Either way, the Punnett square inside will still be the same. So when you set up this Punnett square, offspring number 1 is going to be heterozygous. Offspring number 4 is going to be homozygous recessive. So I tried to color code this for you so you can see here. Now, what you do if you've never done a Punnett square, this capital A goes here and then here. This lowercase a. And then you go down, so this one gets written here and here. So basically, this gets written in every box below it. This gets written in every box below it. This gets written in every box across. This gets written in every box across. And so you look at the different combinations. And so in this situation, you'd have 2 offspring that have thorns, and you'd have 2 offspring that don't have thorns. So if I were to ask you, what is the phenotypic ratio, I would see that for every 2 offspring with thorns, I have 2 offspring with no thorns. So it's really like a 1-to-1 ratio, a 50-50 chance. If I were to ask you the genotypic ratio, you always want to start with what's most dominant. And here, heterozygous and homozygous recessive are 2 options. So that's like a 2-to-2, but we can reduce that down to a 1-to-1. So the genotypic ratio is also 1-to-1. Okay. So here's another example practice problem. You can go ahead and hit pause and check yourself. Okay. And in grasshoppers, so let's go ahead and see if we cross 2 heterozygotes. Let's go ahead and try and figure out the phenotypic and genotypic ratios of these. So hit pause and try and see if you can figure out what ratio this is. Okay, good job. So then when we look at our phenotypic ratio, I'm going to see that I have 3 that are going to have the long antenna, because long antenna are dominant. And then I see that I have 1 that has short antenna. So my phenotypic ratio of 2 heterozygote parents is pretty much always 3-to-1. But now when I look at the genotypic ratio, so for genotypic ratio, I'm going to look at the genotype, starting with what's most dominant. I have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So my genotypic ratio of a heterozygous parent and a heterozygous parent is 1-to-2-to-1.